Ocean. So for those of you who are not aware or familiar with the SAS initiative, the SAS is the Synoptic Arctic Survey. It's an effort for improving data collection in the Arctic Ocean. And what we do is that we aim to coordinate research cruises in the Arctic Ocean in terms of when they take place, where each individual cruise measure and what variables they measure, such that we get a pan-Arctic picture of key data in the Arctic Ocean. The first SAS took place in 2020 to 2022, and I think now some 25 cruises were part of that. We aim to have a second SAS in 2030. So I think there will be sessions at the upcoming ICAP meeting in Boulder next year about this, and this will be coordinated by Shigeto Nishino at the Amstec. Okay, so let's get started. We have three presentations today by Peigen Lin, who is associate professor at the Shanghai Jiao Tong University in China. We have a presentation by Andreas Rogge, who is postdoc at RV in Germany, and we have a presentation by Johan Wegner, who is professor at Umeå University in Sweden. So Peigen, I think we just go ahead with the presentation, and the title is in the slide, so I won't read it out. Okay, Peigen, the screen is yours.
Thank you. Thank you very much. Can you guys hear me clear?
Yes, all looks good. And sounds
okay, great. Thank you very much for inviting me. And thanks for correctly pronounce my university, Shanghai Jiao Tong University. So now, right, I'm working at Shanghai Jiao Tong University in China since 2 years ago, and this study more like when I was done when I was in Wuzhou working with Bob Picard, who is the second author right here, and my other co-authors are Henry and Michelle from the University College London, and Motoyo and Takashi from Jamstec. So today I would like to talk about the recent state transition of the Beaufort Gyre in the Arctic Ocean. So okay. So for today's talk, I will start with a brief overview or introduction of the Beaufort Gyre, and following by my results of the state of the Beaufort Gyre, and as well as the thinning of the cohercline layer. And this schematic shows the circulation in the Arctic Ocean. So the red arrows are the circulation of the Atlantic water, coming through the Atlantic side. And here we more focus on the upper layer circulation, which indicated by the blue areas. And this anticyclonic circulation is the Beaufort Gyre. And this plot shows this from the Prochaski, my colleague in Wuzhou. They made a plot of the freshwater content in the unit meters and for the entire Arctic Ocean. And then you could see this is like the most of the freshwater concentrated in the Beaufort Gyre region or Canada Basin. And this is the largest freshwater reservoir in the Arctic Ocean. And they also find there's an increased trend of this freshwater content in the last couple of decades, from 2003 to 2017, and the increase rate is about 40%, which is really large, and also comparing to the climatology. So this is a really large amount for increasing freshwater content in the Beaufort Gyre. And what is the sources of the freshwater, where it came from, right? And they have a model study shows that there are like two major sources. First is like which shows like the dashed red line right here from the Bering Strait, which is the Pacific origin water that came from Pacific Ocean and not flowing north through the Bering Strait into the Arctic Ocean. And the other main source was from the Mackenzie River, which shows like the thick the red line here, red curve here. And this is the evidence of the Mackenzie River outflow or the runoff, I'm sorry, and discharge. And this is more like a ocean color image from the satellite. And you will see this kind of, high, the yellow colors coming into the Beaufort Gyre region. And just keep those in mind. I will, revisit here a little bit later. So then many study has been aware that the Beaufort Gyre has been changing, and this is the plot showing the edge of the Beaufort Gyre in each year and the core of the Beaufort Gyre in each year. You could see that the gyre is kind of expanding and moving towards the west, the northwest, I would say. So there's this side is northwest, getting close to the North Pole. So and they all in this change and the location shift to the surface forcing and the location of Beaufort High, which is a atmosphere system in the Canada Basin. And the other model study, they did a very interesting experiment. They said the results suggest that if you release the 40% increase of the freshwater in the Beaufort Gyre and within a very short amount of time, and you will see that the entire North Atlantic, the salinity in the North Atlantic decreased dramatically, particularly along the shelf region in the Labrador Sea and the Baffin Bay, and by like minus 0.4 in the salinity. So this is a large change. It could intensify the stratification in the North Atlantic, as well as the kind of weakening or impact on the overturning circulation in this region. And as you know, this is really important for the climate change. And this is why we want to focus on that. And there's two questions we want to address in this study. The one is, what is the state of a Beaufort Gyre in the recent decade, and does it change? And how does the water column respond to the changes? Okay, so right below under the two questions, this is our study region. I want to point out first, and this polygon, magenta polygon covering the region of the Beaufort Gyre. So our data will average within this polygon in the following results. So first of all, I will show you the time series of the dynamic ocean topography from 2003 to 2019. So the dynamic ocean topography, you could consider this as a reflecting the state or intensity of the Beaufort Gyre. So it's higher dynamic topography represented the stronger Beaufort Gyre or expanded Beaufort Gyre. So as you can see that overall there's an increased trend from the 2003 to 2009, but you can also see there are notably variations between the trend. Then we divide this time series into like two periods. The early period from 2003 to 2011, and the second period from 2012 to 2019. And you make the trend, you see that there's strong increase in the early period, and the increase rate is kind of become considerably slow in the second period. So and we talk about the freshwater content, right? Then let's look at the freshwater content right here. And we made or constructed a time series of freshwater content as well during this time period, and you see that the freshwater content increased dramatically in the earlier period, and it becomes more stable in the second period. And I would like to know there are two freshwater content time series, right? So we actually did compute the freshwater content in using two different data set. The magenta curve, we were using the historical hydrographic data within our study domain that I showed you a second ago. And the other, we use a DOT, which is the dynamic ocean topography plus the GRACE, which give us the equivalent water thickness. And we can also estimate the freshwater content. And interestingly, by using two independent data sets, we got really similar results, which give us the confidence that the trend does make sense, that they have increased dramatically in the earlier and gets stabilized in the later. And these two time series, and this is also associated with the DOT variations. And what is the spatial change looks like? So then we constructed the map of the trend of DOT in the earlier period from 2003 to 2011. And you can see that the part is positive values or positive trend, which means increased trend in over throughout of the domain, and particularly the peak, the maximum is located in at the northwest of the Canada Basin, and this is consistent with the fact that I just described a second ago, that the Beaufort Gyre extended to the northwest, right? And this is what the previous studies they found the fact. And by looking at the freshwater content in the same period, and we'll see kind of really similar pattern, and it picks up at the northeast part or northwest part of the Canada Basin, and if we do the same calculation for the later period, and this is the trend for the DOT, now the thing changes, and from 2012 to 2019. And you can see the increased trend more located in southeast of the Canada Basin, and to the other side is decrease. And there's a kind of similar pattern shows in the freshwater content as well. So then we made a map of the trend of wind stress curve, and you can see that the wind stress curve is negative trend, and in the later period, it's negative trend where you have an increased DOT and freshwater content. It makes sense dynamically, right? When you have like more negative wind stress curve, you have stronger Eichmann pumping, and which leads to all these kind of changes. And to the other side, the decrease of freshwater content and dynamic ocean topography kind of likely associated with the positive wind stress curve on the other side. It's this dynamic link kind of also consistent with what the studies shown before. And this is the spatial pattern, and we want to address the second question, which is how does the water column respond to the changes? Okay. So now let's return to the time series I showed you before above, and now I added one more time series, which is the thickness anomaly referencing to the first year. So it's more like a thickness in the upper layer. So what I mean about the upper layer, so those are the mean profiles of salinity and the potential temperature within the Canada Basin. And it consists with upper layer, the surface layer, the warm helicline layer, the cold helicline layer, and on top of the Atlantic water layer. So now we more look at the layer above the Atlantic water layer. So consistent of the surface layer 1 kilocline and a cold kilocline. So you see that the time series or the variation again looks really similar to the freshwater content and the DOT, right? And dramatically increasing in the early period and it gets stabilized in second period. And if we look at it into details, and what we do is we compute the trend of the volume of the water in each salinity class for the early period. And it consists with the warm kilocline and cold kilocline. Salinity ranges from 28 to 34. And apparently the trend are positive, particularly when the salinity larger than 30, and the peak that right above the cold kilocline, which is shading here. And this is the early period, and what looks like in the second period. The later period, you see the majority or the negative trend occurs in the cold kilocline. This is the biggest change in the second period, and this thinning of the cold kilocline is the main reason of the stabilization of the total thickness in the later period. And now we want to address what's the reason, right? And to address that, the first thing coming into my mind is we should look at the Pacific origin water first. Because as I said in the introduction or in the very beginning, that the Pacific origin water that contributed to the freshwater in the Canada Basin. So this is a schematic of the Pacific origin water circulation in the Chukchi, and it flowed was northward along these three main pathways on average, and the majority of the Bering Strait inflow drains off the shelf through this so-called Barrow Canyon. And once it gets off the Barrow Canyon, it turns to a small portion of the Pacific water that turns to the east, join to the Shelby Jet, and the majority part of Pacific water turns to the west and join into the slope current. So they all have a chance to get into the Beaufort Gyre. So now what we do is that we find the mooring data right at the mouth of the Barrow Canyon, the mooring data provided by our James Tech colleagues, and we can do the kind of similar calculation to compute the trend of the volume in each salinity classes, right? And when we do that, and this is the first period, and this is the dots right here are showing the result from the mooring data at the mouth of the Barrow Canyon. Okay, and then you will see that the trend is quite similar to what the trend found in the Canada Basin, within the Canada Basin, right? And it also peaks a little bit above the cold kilocline. And what looks like in the second period, and this is a second period, and you see that the red dots kind of a little bit follow also and above the cold kilocline, and it turns to be the negative in the cold kilocline, right? And there are two take-home messages, right? The first is the Pacific water does have the similar trend shape, comparing to what the trend in the basin. And second take-home message is the trend of the Pacific water is a little bit less, well, not a little bit. It's like a significant less, because this is the range, confidence level, and in the magnitude, particularly in the later year, right? The first year is okay, but the later years, they are significant different. So we were expected to see there probably the other sources could probably contribute to the trend in the second period. So then we're looking for the local formed water in the eastern Beaufort Sea and on the other side. To adjust that or investigate that, we had two experiments by using the Lagrangian particle releasing in two like Lagrangian particle experience. The first one was done for the 2011, which is the last year of the first period when the Beaufort Gyre located here. So when the Beaufort Gyre is more located in the northwest. And the second experiment was done for the 2019, which is also an extreme year when the Beaufort Gyre located in the southeast of the Canada Basin. And we have the particles initiated along our 100 isobath, and the blue dots or blue particles are from the Churchill and western Beaufort Sea side, and the red particles are released on the eastern Beaufort Sea. So and we run it for a year for each of the experiment, and let's see the result. Okay, then you see that in 2011, and like most of the particles, blue particles kind of gets into this Canada region, the Beaufort Gyre region, but the red particles are more like dangling, around the shelf, not really getting to the region. By contrast, then in 2019, like more red particles are get into the Canada Basin or Beaufort Gyre region, right? So we wanted to quantify that the percentage of the particle into the Beaufort Gyre, the Churchill Sea and the particle from the Churchill Sea and the Western Beaufort Sea, there were 90% in 2011 get into the Beaufort Gyre, and only 15% of the Eastern Beaufort Sea get into the gyre. But what about like in 2019, the number for the Churchill and the Western Beaufort, it decreased a little bit to 84, but to other side, it increased dramatically, significantly to 73% in the second year. So then I would like to note that the water, the water in the Eastern Beaufort Sea is fresher than what the water to the other side, to the west, which means you have less contribution to the cold hill climb in the second period, which could explain a part of why it's thinning the cold hill climb in the second period. So all these processes are due to the shifting of the Beaufort Gyre. So here I would like to sum up. So during the last decade, the Beaufort Gyre has transitioned to a new state, the shifting towards the southeast of Canada Basin, where the active winter curl has intensified, and together with a deepened rate of increased dynamic ocean topography and a stabilization of freshwater content and thinning cold hill climb layer. And the cold hill climb layer has thinned significantly due to the less input of the cold and salty water from the Pacific water and Churchill Sea shelf as well, together with a greater entrainment of lighter water from the Eastern Beaufort Sea as a result of the southeastward shift of the Beaufort Gyre. And this Beaufort Gyre state could be a precursor of larger freshwater release, particularly potentially threaten the overturning circulation in the North Atlantic. So this study has been published in the Nature Geoscience in last year. And I will stop here and take any questions. Thank you, guys.
Thank you so much, Peigen, for a very nice presentation and clear presentations. people feel free to ask questions, but raise your hand before so. I can maybe start with one, Dan. You had a very, I was wondering about, your final conclusion point. Whether this sort of signifies a change in the stability of the gyre and freshwater storage, and apparently it does. Maybe you can elaborate a bit more on that point.
Oh, what kind of point? I'm sorry. Can you
Yeah,
can you
so
repeat?
this just said that this could be a precursor of a large,
Precursor of this?
yeah,
Okay.
freshwater release. Can you elaborate a bit more on that?
Oh, this good point. So we are thinking because as I said in the very beginning that freshwater content increased by 40% in from comparing to the climatology, and now the freshwater content kind of stabilized in the recent decade, and this is just our suspicion that since the water not changing but not changing in the Beaufort Gyre region, but the inflow of the Pacific water is still increasing, and also the other sources are also increasing. So which means that you have more fresh water released somewhere in the Arctic Ocean through the kind of archipelago or the front strait to North Atlantic, and you will expect to see more freshwater export from the Arctic to the North Atlantic that could impact the overturning circulation in the probably near future. This is our we don't have the evidence yet, but yeah.
But is it possible to look at the structural stability of the gyre? Is that somehow something which can be calculated?
Yeah, so Mary Louise Timmermans and John Marshall, right, John Marshall, they have a review paper about the freshwater content in the Beaufort Gyre, and they report a 2 more year of of the time series that they do see a decrease of the freshwater content in the Beaufort Gyre, and but we don't really have the change, like significant change of the outflow in the front strait for now. But I guess they may take a little bit of time, to circulate to the front strait and kind of archipelago.
Yes,
yeah.
I guess we are. But there's certainly observations in the front strait which would capture the outflow when it comes. Okay, we have a question from Annika. Annika, I can read out the question or you can post it yourself, but then you need to open your microphone.
Okay, can you hear me?
Yes.
Really nice talk, thank you. I just had a question about your DOT analysis. I was wondering what motivated you separating it into your year groups of 2003 to 2011 and then 2012 to 2019, and how sensitive your results with the trends and the spatial analysis are to those year groups. Thanks.
Thank you for the question. This is a really good question. So we did several sensitivity experiment for dividing how to divide the time series. So first of all, we do like 4-year running trend calculation, and then we see that when there's the minimum trend was found in 2011 to 2012, this region, and this is first. And then the second is we change the dividing part of dividing year to like 10 or 9 or plus minus 2 years. So it doesn't change very much for our result. And also the freshwater content kind of support our division right here, right? Because they're showing the stabilization since 2012 versus 2011. So that makes us quite confident about the result. Hope I answered your question.
Yes, thank you.
Thank you.
Okay, then we need to move on. Thank you again, Peigen, for a
Thank you.
really, really nice talk. That was very interesting. And we move on to Andreas Rogge at the RV. Andreas, you just share your screen. And the title is, I can read it out now, Dense Shelf Water Propagation and Subduction Drives Lateral Injection of Particulate Organic Carbon into the Deep Nansen Basin. Okay, Andreas, take it away.
All right, hello everybody. I hope you can see it and hear me as well. So the title is pretty long. Next time I will shorten that a bit. So first of all, thank you for the opportunity to present my work here. That paper was actually quite a while ago, so in 2009, no, I don't know. So for quite a while ago, which is the reason why I will also show you a little update at the very end. So there are some more progress in that work. And this work was done at University of Kiel back in the days. And right now I kind of switched kind of to the dark side and I'm working in Antarctica right now. So yeah, it's been a while. So first of all, I will give you, can we remove that part here up there? Do you see that bar up there?
No, not before.
So it's only me you see, so that's okay. So then first of all, I will give you a short introduction about the biological carbon pump. Very briefly, so in the upper ocean in the euphotic zone, we have the production of POC, so by phytoplankton, and this causes a decline in the concentration of dissolved inorganic carbon and causes the concentration gradient towards the atmosphere and the influx of CO2 into the water. And because POC is sinking above a certain size, it sinks through the water column. But usually the POC gets remineralized back into the DIC pool and recycles back into the atmosphere. And only that part which can kind of sink through the pycnocline and end up in the deep water can stay there for a pretty long time, which is what we call long-term sequestration. And this whole process is causing a pattern in the POC concentration, which is usually described as an exponential decline with depth. So this is POC flux, which was first described by Martin et al. In 1987. And the POC concentration is usually considered to be within the same pattern, so also this exponential decline. But Martin already knew that this model is actually highly simplified because of the obviously stringent assumptions imposed by ignoring lateral particle advection. So I like the way they wrote it back in the day. So basically, he's just considering vertical export and not some lateral transports. So, and then a little introduction about the transpolar drift for you. So what we have in the Arctic is the feature that we have steady winds from the Eurasian continent, which pushes the sea ice towards North America. And this is causing open water conditions on the Siberian shelves, which then lose more heat, which increases the sea ice production in the beginning winter. And because of that, we have cooling and brine rejection from this feature, and this causes the formation of dense shelf waters. So in particular, Barents Sea bottom water, which is built up here, and what I want to talk about today. So, and the last introductory slide is about the inflow of Atlantic waters. So in Fram Strait, Atlantic waters are entering the Eurasian basin and form the boundary current. But one branch also enters the Barents Sea and brings in nutrients. And together with the open water conditions there from the transpolar drift, we have an increased productivity in the Barents Sea. And the Barents Sea bottom water, which is formed there, is crossing that productive area and then subducts at the continental slope. So it leaves the Barents Sea here in the St. Anna Trough, which is a depression, and then it propagates below the Atlantic core of the boundary current towards east. So the question for me was then, what is the impact of this interplay of dense water propagation and biological productivity on the biological carbon pump. And therefore, I joined a cruise in 2018 on board the academic Trozhnikov. We did a lot of CTD profiles, including fluorometer and turbidity. But also we had an underwater vision profiler mounted on the CTD, which is basically, so it's this device here, and it's basically a particle counter. So in that main cylinder here, you have a camera, and you have two light units, which light is certain defined volume of water. And it counts particles above the size boundary of 100 microns and takes images of each particle about above the size of 1 millimeter. And additionally, we took size fractionated samples for POC and DNA, so larger volume above 100 microns and a smaller volume for the suspended fraction below 100 microns. And when I processed the raw data on the ship, I saw already that there is actually not really this exponential decline. So there was a mid-water plume of particles. So this is the UVP raw data for certain particle size classes. And this is what I kind of expected in the Arctic Ocean production in the surface, and then an exponential decline towards depth. But that was obviously not the case. And this profile appeared repeatedly in the Nansen Basin, all along the continental shelf. And if we put together our data, we see that here I will right now talk about that transect only because here we have most of the data. So in that transect, which is also closest to the outflow region in St. Anna Trough, we have a plume which expands about 40 kilometers into the Nansen Basin at about 1,000 meter depth. And this plume of particles was highly associated with Barents Sea bottom water. Although a smaller fraction of particles was already kind of sinking out. You also see that here in the section plot. The black isoline is the salinity and the dashed line is the density. So in between those lines, you have the Barents Sea bottom water, and the fraction of the particle load actually sank out of that plume. So first of all, our question was, where did the particles really come from? And therefore, we have some evidence from our field data. So we see, as I just told you, a sinking separation. So the larger the particles get, the less associated they are with the Barents Sea bottom water. And this means that the particles needed to have a long trajectory because sinking takes time. So you can assume that they are kind of coming from a distant source. Next, we also saw in the plume an elevated signal of chlorophyllescence. And in the UVP images, we also saw that we have a lot of grazers there, so especially copepods. So it seems that we had a bloom community from the euphotic zone there. And we also saw in our DNA data, so this is a PCA plot, which shows you the community differences from the different samples. And so I split that up into prokaryotic communities and eukaryotic communities for the suspended fraction and the sinking fraction. And you see that especially for the suspended fraction, the microbial communities, both prokaryotic and eukaryotic, were distinct from each other. So this also points towards non-local material, so that there's an intrusion from somewhere else. And this difference in the size fractions we also saw in our CN ratios. So the particle fraction above 100 microns actually had a very high CN ratio, which points towards pheocystis, so a high tap load, so transparent exopolymeric particles, which contain a lot of polysaccharides. While in the smaller fraction, those are probably mostly diatoms and heterotrophic flagellates. And as the last point from our field data, we saw that from the UVP images of the detritus, that most of the detritus, so this is above 1 millimeter now, it's only large particles, but they consisted mostly of compact stuff. And those compact particles only appear when your POC is going through different levels of the food chain, so it's highly densified, and this points towards degraded material. So altogether, this points towards older material from distant sources, And now we wanted to know, is it really shelf-based? So we applied a model, the so-called FISA model, finite element sea ice ocean model, which is a numerical ocean model. And we did some particle backtracking. So this is again our transect close to St. Anna Trough here. And you see already that the main current field here from the Barents Sea is crossing our transect. And then we released particles in the dense water there. And we saw that here is the backtracking. So the color code shows you where the particles reached the surface in the backtracking, so where they're formed. And 50% of the small fraction there originated south of 81 degrees north, so on the shelf. And the trajectories were on average about 750 kilometers far away. However, one needs to consider that resuspension is not included in the model. So if we have dense water leaving the trough, you can assume that there is a lot of mixing on the bottom. So also resuspension is going on. So I assume that the trajectories are likely longer in reality. So together, like the shelf origin is promoted by the model, I would say. But as a next step, we wanted to know how much POC is really sequestered from the shelf. And therefore, I applied two simple regression models to estimate the POC from the sensor data in order to get a high resolution data set of POC. And for the fraction smaller than 100 microns, so this is the suspended fraction, I used the chlorophyll fluorescence and the turbidity. And for the fraction larger than 100 microns, I use the particle size distribution from the UVP as well as the total particle volume above 100 microns, of course. And together, this looks like this. So you see clearly this POC smaller than 100 microns, which is associated with the dense water. Larger than 100 microns sinks already a bit out. And the total POC in the plume was about 27 milligrams per cubic meter, while about 60% of the total POC load was made up by the small fraction. However, oh, yes, no. Okay, we want to have fluxes. So concentrations is not the only thing. So we also need current velocities. And luckily, in that transect, that was a mooring array, which is also the reason why we have those full depth profiles. So there were rotor current meters and acoustic Doppler current profilers deployed for about 3 years. And the average current velocity of the dense water on the day of sampling was about 6 centimeters per second. So if you multiply that current velocity with the concentration and the area of the dense water, you get a flux, a lateral POC flux of about 2.3 kilotons per day. And this equals about 8.2 kilotons of CO2. And I like that number. So it's if 1,000 cars are circling the globe per day, that's the emission which kind of gets sequestered there. Or 3 coal-fired power plants, but that number is rather small, so I usually prefer the 1,000 cars. However, you have to consider that this is not the shelf slope or the shelf break, right? So there are losses from the Anna Trough towards our transect, and therefore, like to really quantify the outflow from the shelf, we applied another model, the biogeochemical model RECOM, which is the regional ecological model. So it consists of phytoplankton, racers, microbes, and so on. And it's built on top of the ocean model. And first of all, what we saw is that this model is reproducing the plume of POC in the dense water. So this is our real transect again. And this is the model transect. But the concentration was much, much lower. So it was like 10 times lower. And also the depth was shallower because the dense water in the model is less dense. So, and therefore we applied a correction factor for the POC. So we just multiplied everything in the model by about 10. And we included the seasonality from the model. So this is a very complicated plot, I need to say. But the thing is, what you see here, this solid orange line is the outflow out of St. Anna Trough. And you see that there's a seasonality. So this is the summer production, of course. And only if you have the summer production, there can be some outflow. So we included this seasonality and integrated then over the whole year, applying our correction factor from the field. And doing that, we get a total outflow from the shelf of about 1 megatons of POC per year, which is about 3.6 CO2 equivalents. And I like that plot here, which is a three-dimensional visualization. So you look actually from the North Pole towards the Siberian shelf, and here's our Sustenance trough. And our POC is leaving the shelf, and here we have our first transect. And what I wanted to know is also what's happening with that material. So first of all, okay, I forgot that point. So just to put that number of 1 megaton into perspective, this is about 30% of the whole burial on the Barents Sea. So it's really an increase in long-term sequestration if you assume that the deep water circulates for a pretty long time. And it's also an additional food source for the Arctic deep sea fauna, this process. But as I said, so I wanted to know what's happening to that material, and then we can look into our other profiles or the other transects we have. And first of all, like our first transect again, so we already saw that large particles are sinking out of the Barents Sea bottom water. So you see that here, it ends up in the upper polar deep water. And if we take a look at the next transect we have in the Eurasian basin, we see that the plume is expanding. So like the water is kind of getting distributed above the upper polar deep water. And we see that the Barents Sea bottom water signal is also decreasing over distance. So that little nose here in the polygon is actually getting smaller and smaller until it's vanished in our last transect here. But you still see that there is a certain particle load, right? So, and this is only the particle load which ends up in the upper polar deep water. So it's a remnant of this injection into the Nansen Basin. And for me, that story was actually over now. So I shifted over to Antarctica, got a new job as a postdoc. And so that story was over for me. But then I received a call from my dear colleague, Barbara Niehoff, and she's told me that, oh yeah, there's something in our SAS Odin data from 2021. So maybe this is interesting for you. And well, it was. So the thing is, across the North Pole, if you plot the UVP data, which they also deployed there, you see that there is an intense plume of particles as well below about 2,000 meters. And with a horizontal dimension of about 150 kilometers. But of course, the concentrations are lower there because it's more far away from the productive area on the shelf. And it's associated, when I was searching the literature, this is actually from the same cruise here, we see that this plume is associated with Eurasian Basin deep water. So it's not only in the upper polar deep water anymore, but it's in the Eurasian Basin deep water. And this little nose here are actually the two stations behind the Lomonosov Ridge, so in the Makarov Basin. So there we see there is no increase in particles at all, which I find kind of interesting. So I was digging a bit more into that, and I was also looking into our mosaic data we had, which was in winter, and there we also see an increase in the deep particle concentration. So here you see those left stations are our stations from our Arctic 2018 cruise. And those are the new stations from Mosaic, and you see that there is something also going on here. Yeah, and it's further away from the Lomonosov Ridge than the Odin data. So this is also why the concentration is lower here. So if we put that all together here in our map, so this is the mean particle concentration between 1,200 and 3,500 meters. And I already excluded the nebuloid layer, so without the lowermost 300 meters of each profile. And generally, there is an increase in the deep particle loads close to the Siberian shelf, of course, because that's where the dense shelf water is subducting, but also close to Lomonosov Ridge. And the closer you are, the higher the particle concentrations get. But it's not in the Makarov Basin, as I told you, and this is because the deep convection here in the Makarov Basin is going the other way around. So it's not coming from the productive Siberian shelves, it's coming from the less productive area around North America, where we have the high sea ice concentrations and also no nutrient influx from the Atlantic waters. So if we, that's my last slide. So if we plot our dense water, so those are the TS plots only on that, about that tail there and for the dense waters. And the x-axis is the distance from the Siberian shelf. So along that transect here from the Siberian shelf. And the y-axis is the distance from the Lomonosov Ridge towards, well, let's say Franz Josef Land, into this direction. Then we can follow the circulation of the Eurasian Basin deep water, actually. And we see here, this is our transect again here, where we have the Barents Sea bottom water and that injection. And we see with distance that the particle load is kind of get transferred into the upper polar deep water. This is what we already know. That plots here, they are a bit out of the thing, so it's not really final yet. And then we see here, this is at the Lomonosov Ridge, this is this station. So there is another dense shelf water intrusion, maybe from the East Siberian Sea or so, but we still have the particle load at the Eurasian Basin deep water. And if we follow that, we see that also here in that TF plot, for example, or in that's from Mosaic and from the SAS Odin cruise, but we also see it here close to Greenland, where it's kind of the water is circulating around. And we even see it at the exit close to Fram Strait here, that little nose. So this is kind of still work in progress, but I find it really astonishing that we see kind of a process which is transporting a certain particle load all around the Eurasian Basin. So yeah, just what I showed you, there's a transfer from the different water masses towards the Eurasian Basin deep water. And my question is actually what I still need to figure out is how is the process working? Why is the stuff not sinking out, or why is it not remineralized? And is it really a permanent feature because we also saw it in winter? So it is, to wrap it up, we kind of found a large-scale transport structure in the Eurasian Basin which carries a certain particle load. And this kind of means that lateral transport is more important for the deep sea in the Eurasian Basin than previously considered. So it's not only the vertical transport, there are large areas where we have an inflow of particles from distant sources. But the detailed process understanding and also the impact on the carbon supply, so the quantification is still lacking, and I'm working on that. So that's the status now. And with that, I want to thank you very much.
Thank you so much, Andreas, for a fascinating presentation. Awesome work. We have open for questions, so anyone write in the chat, so raise your hand. I can maybe start to ask. So you would recommend then for the next SAS that all of the ships should carry UVPs on their CTDs, I guess.
Well, always, yes.
Always. Yeah.
Oh, well, the thing is, it's easy to have, right? So you don't need to sacrifice bottles or something like that.
No.
You can just mount it behind the bottles.
Yeah.
It's a nice piggyback sensor which produces a lot of data, which is really, really good. So.
Yeah.
And especially, so this is what I haven't done so far for the other data, is to include also the beam attenuation, because this is not intercalibrated. And this is also kind of a good thing about the UVP is that every sensor is intercalibrated with each other. So you can really intercompare all your data. So yes, so short answer, yes.
Okay. Nice. I'll remember that. Also, I wonder, is it possible to sample the material and determine the age? So you know how old that POC is? That would be interesting, I would think.
Yeah, I would actually love to do that. So that's also why I wanted to use that round kind of a call. So if you have some more data about that, so also on POC, but the ages as well, it would be really, really good to have that. But for 14C, I did that in Antarctica, and so you need a lot of sample volume, of course. Yeah. So I mean, even for the POC concentrations, we were sampling about 20 liter for the fraction above 100 microns, right? So, and for 14C, you probably need even more. So, but I think it's worth a try, definitely.
Yeah, I think it would. Yeah. Johan, please go ahead.
Yes, thank you, Andreas. Very interesting with these intriguing particles. I just wondered if, I mean, do you think it's biological material, and is it possible to sample it and do kind of experiments and to look about the potential remineralization or other aspects of that, or have you done that, or somebody already?
So I hadn't done that yet. So I think it's all, as I said already, it's a matter of your sample volume which you can get. Because the thing is, I mean, in my plots there at the North Pole, it looks a lot, but actually I kind of adjusted the color bar a lot. So the concentrations are rather low, but if you assume that it's a permanent process, I think the impact on the carbon supply is pretty large over time, right? And talking about experiments, yes, I think it would definitely be worth it if you get the material. So the thing is that I was showing volume concentrations. And if you would plot the particle numbers, so the numerical data only, you would see that those larger particles, there's maybe about 1 to 2 particles in 1 cubic meter. That's not a lot. So you really need a lot of sampling volume, or you really focus on the suspended fraction smaller than that, but I don't know what the load is yet. So, but I think it's definitely it's a feature which is kind of overseen. So we know that we have a circulation there, and we also know that DIC is sequestered with those water masses and it's enriching in those deep waters. But now it seems that also suspended POC, which makes sense logically, is also circulating with those current there. And I think it's definitely worth to investigate much further. So I didn't have the chance, but maybe one day.
Well, thank you. Some large filtration equipment perhaps is needed for the next SAT expedition, right?
Exactly.
Okay, any more questions to Andreas before we move on? This is the final chance. There's a question from Paul Berkman. Paul, would you like to state your question yourself? Then please just open the mic and go ahead.
Good morning, everybody. Yeah, just an observation. The increase of freshwater content in the Beaufort Gyre appeared to be until around 2012, which was when the absolute minimum of sea ice occurred in the Arctic Ocean. And there had been an increasing frequency of sea ice minima that were recognized and pointed out from about 2007 to 2012. So I'm just wondering, is the Beaufort Gyre acting as some sort of pump on a circumpolar scale for the sea ice?
I think we need to direct that question to Peigen. Are you still with us, Peigen? Maybe you can just answer it.
Yeah, I'm still here.
Yeah.
I think this is still a really good question, and seems so for we don't have observations, but the model study shows that the ice melting is not the major source of the freshwater content in the Beaufort Gyre, because the two major sources I just said that are Pacific water and the river runoff, because the I'm not quite sure how that links to the ice melt to the freshwater increase.
Well, I'm not actually thinking that the ice melt relates to the freshwater increase as much as the freshwater increase in the circulation of the Beaufort Gyre in some way are pumping the dynamics of sea ice on a circumpolar scale.
Yeah, I'm not quite yeah, this is a good question. I should think about more about this. Thanks.
Certainly.
All right. Thanks for the question, Paul. And thank you, Andreas, for a very nice presentation. And we move on. And now it's Johan Wegner from Umeå University. Contribution from prokaryotes to the Central Arctic Ocean carbon cycle. So, Johan, please, the screen is yours. We can't hear you, Johan.
So, sorry, now I find the control disappeared when I put on the screen. Can you hear me now?
Yes, we can hear you, and the slides look awesome. So just go ahead.
Yeah, we start there, I think. Okay, thank you very much. Andreas brought us to carbon and said something also about the SAS Oden expedition that was arranged by Sweden 2021, and I will expand on both. My title is the contribution from prokaryotes to the Central Arctic Ocean carbon cycling, and that describes a little bit the aim with our own work package. And prokaryotes here are then bacteria and archaea, of course. You can think of them as bacteria, both of them, but they are quite distantly evolutionary related. The picture or photograph here is from one of the bright days when we did some ice sampling, and we were happy about that because we were in polar bear territory. Oden, it looks like this. It's Sweden's largest icebreaker, about 100 meters long, and it's an excellent also platform for research when it's not helping cargo ships in Swedish water during winter. So we were very grateful that this opportunity could be arranged, and this is from another ice sampling, I think somewhat north of Greenland. We were like 75 people on board. Well, 38 of those were scientists and 22 were crew members, and 15 were from the Swedish Polar Research Secretariat. Extremely valuable support for us scientists. Now the expedition route looks like this. Something happened again. We went from Sweden here past Svalbard in the Pram Strait, and then turned a bit right and went straight up towards the North Pole and turned down the Lomonosov Ridge, and then back again north of Greenland, and then back towards Svalbard. And to the right you see the route in more detail with the sampling stations, and it was about 2,900 kilometers. We spent 43 days in the ice, about 2 meters thick. So we're grateful to the crew that they could take us all the way around the trip. The red dots there is pelagic stations. So when you see a white star, it's on ice stations. The blue dots are Efika fishing stations, where they made some vertical trawls and looked for fish stocks and their size. And there's some white dots in some of the dots as well that you may see, that was sediment core sampling. So quite a lot of work was done during this expedition. The Synoptic Arctic Survey, if we should remind ourselves some of the aims that I summarize a bit briefly here, it's of course to investigate climate effects then on ecosystem productivity. That is something I think we contribute to with our work package, hopefully. And not so by diversity, but by diversity is also one important question to see how is that changing. Well, the Arctic is turning more like this. You see in this photograph, it was quite a lot of open water northeast of Greenland at the time that we were there. Also, as I said, we want to know if there are some fish stocks there worth of utilizing commercially, and if they will become more available with time. And also, of course, capacity to buffer the capacity of the Central Arctic Ocean to buffer for CO2 and methane, and perhaps we contribute a little bit to that question, perhaps less to the resistance to pH decline, which was another major question addressed by SAS. So we'll see. Hopefully together we can at least give better answers to this in the near future. Just to present a little bit what was done during this expedition, it was 16 work packages. I was told this was unusually many biological work packages on this expedition, which was fun to be part of. And the biological one, VP1 to 8, looked at the different aspects of biology from viruses actually to fish and prokaryotes and zooplankton in between, and zooplankton of all different sizes. VP9 was focused on paleoecology and took this sediment course and got some amazing records 100,000 years back. So hopefully we get some very interesting information. And you see the expedition leader, Pauline's nice Leonmalm in the nice red outfit in the back there. Also, we see work package 10 and 13 that looked at chemistry that had to do with ventilation of the Arctic basins. And the oceanographic packages looked at more mixed layer depths and also ventilation. And then even one meteorological work package that wanted to better facilitate modeling of the Arctic climate. And it was apparently too little meteorological data available for that at this time. If you want to know more about these and who is possibly the data owner, you can go into the Swedish Polar Research Secretariat homepage and find the report of the expedition. So please do that if you want to collaborate, and we really would like to do that, and hopefully we can find some good projects to synthesize good papers in the end here. But let me go to our work package, where I can mention a little bit about the few preliminary data that we have and what questions we addressed. And we that work with this work package is my postdoc Ashish Verma to the left, sampling on board Oden, and myself, and then Jeroen Pinhasi, that is the professor at the Linnaeus University, and his previous graduate student, Dennis Amnebrink, that was also helping us with the questions and the sampling and measurements. He's now had his dissertation, so he's a doctor at this point. But the questions we addressed then in this work package that we call adaptive strategies of Arctic prokaryotes is how much of the primary produced and terrestrial carbon entering the central Arctic Ocean that is metabolized then by prokaryotes in the water column. How much are they contributing to the cycling of carbon? And in that perspective, it's important to have a good estimate and know how efficiently they transfer the carbon they assimilate to biomass. The alternative, of course, mainly to transfer to carbon dioxide and release that. And also, we want to know if this growth efficiency is then regulated by, for example, temperature or substrate supply primarily. Well, the basis for this is to try to contribute to improve the model we have of the carbon cycling of the Arctic Ocean. And there is a couple of efforts that have been done and developed good models for this, but they are focused on more parts of the Arctic Ocean, specific sea areas. And also, we think the growth efficiency estimates are not too well constrained. We have one model to the left there for the Barents Sea, which was published by Watzmann et al. 2006. And when we looked at the prokaryotic growth efficiency they used there, it was 0.5, and 50% of the assimilated carbon can then be transformed to biomass and half to carbon dioxide. That's a little bit high in my perspective if you just consider the current estimate for the oceans in general, which is more like 10%. So it might be a need to look at that and possibly revise that. In the Amundsen Gulf, it's outside the Canadian coast. More recent modeling effort by Forrest et al. Has used more like much lower values, 0.07 to 0.25, which is closer on average to this 10% level of growth efficiency. So that might be more in line with that. But again, this is done for a more limited part of the Arctic Ocean. Interestingly, they found in that study that as much as 53% of the secondary production or cycling of carbon in that area was due to microorganisms. So quite a large part of the cycling can be due to the microbial activities. The preliminary lessons we learned from this study now is that we actually have the highest biomass growth and respiration is in the cold 50-meter upper surface layer. It's also very low both biomasses and process rates compared to other more temperate environments, of course. than we found in the temperate zone, then, for example. Also, we did, I think it's not done too many times in what we found in literature, respiration measurements, and can thereby determine the growth efficiency up in the Arctic, Central Arctic Ocean, and it was really low. It's more like 2%, and values 10 times higher than that has been used in the literature recently. We also could demonstrate, despite this quite little small data set, relationship between growth efficiency and chlorophyll A, which would then say it's more like substrate supply than temperature that is important to regulate the growth efficiency. So let's look at a few data supporting these insights. This is show the prokaryotic abundance in cells per liter times 10 to the 8, and we have depth, and as you see, we sampled with this in our work actually down to 500 meters. We can do. Of course, we have much deeper water column in many areas, but this is what we can manage, and especially for the activity measurements, we're not sure we can really handle pressure effects and so forth, since we can't incubate the samples at the realistic pressure. So we settle with going down to 500 meters. It's 13 profiles that we have in a larger data set, and you can see it's low in the respect that usually we present this data as a 10 to the power of 9, not 10 to the power of 8. So it's about 10 times lower, but it's also clear that it's in the surface layer, upper 100 meters. It look like here that we have the highest abundance, and also they go a little bit deeper. The high abundance is in the closer to the ice edge, in the North Pole area and Macro Basin, around root distance 1,000. You see the shallowest depths of biomass distribution, but so it's a little bit different spatially in that respect. There's also low prokaryotic activity. We can look at the data more in depth profiles like this. And if we look to the left in the panel, the blue is the temperature. And you see they have a little bit higher temperature, but it's still, the scale is down here. It's around 1 degree here from 5 meters and upwards, and then it decreases, and around 150 meters, we go below 0. And up in the upper 100-meter layer, we have something like minus 1.7 degrees Celsius, close to the freezing of seawater. So of course, it's quite amazing that these creatures can actually live at these cold temperatures. As Peigen showed in his presentation, I just noticed that it was a little bit similar shape in the curve. We have a salinity around 34, as you see up in the upper scale here, up to about 100 meters, then decline. So it's almost like 4 full salinity units pressure in the surface, and it is 100 meters deep down. So these are the characteristics, and then you see again that prokaryotic abundance, as I said, we have highest in the upper, at least 50 meters here, and the same is true for the growth rate of the prokaryotes and also the respiration rates, and we have really low rates both for the growth and respiration again here. Colors here just represent the different sampling stations, though. Just to give you some perspective, we have some experiment with our measure values we've taken ourselves, go outside the Swedish coast in the Atlantic here, and just to give the impression how low the values are, actually, except for I think 1 point here, it's a cluster of values very close to 0 down here. So for the growth rate here, we have like 10 to 40 times higher values, or in the Atlantic than in the Arctic. And for the respiration, it's more like 2, maybe not so much. When you have values here that's below the pycnocline in the Atlantic, these values up here is above the pycnocline, the photic zone, and of course they are higher in August, which was about the same time as we did the sampling in the Arctic. So they're really low samples, close to the detection limit of metals to try to measure this with the current methods. To give a perspective of other studies, we actually found one study that passed the North Pole and both Macro and Amundsen and Nansen Basins, and also did measurements of prokaryotic biomass production here. And that was Wheeler et al., published 96. I think they were up there 1994. And as you see here, actually, we have about 3 times lower rates than they have on the values they published in their paper, which was some averages for different latitude sections. And now this is entirely actually explained by lower thymidine conversion factor that we measured, actually determined empirically up in the Central Arctic Ocean. Wheeler et al. Assumed the conversion factor from literature data that was mainly derived from lower latitudes. So that could entirely explain that. So actually, if we apply the empirical factors we determined now on Wheeler, our measurement was much closer, maybe not significantly different, but actually we're 47% higher than Wheeler's numbers. So that would revise those a little bit if they are true. So by measuring the respiration by prokaryotes, we could also then determine the growth efficiency and actually find the statistical relationship with chlorophyll A and also the prokaryotic growth rate itself. But primarily, you can see on the y-axis here that they are really low. They are being, say, less than 0.1% to about 5.5%. It's really low values. And recent reports have used about 10 times higher values, which is common in other sea areas. Perhaps not too unexpected, since the really oligotrophic conditions and cold temperatures may demand a high maintenance activity by the prokaryotes also to manage that environment. So the transfer of carbon to biomass will be really low. The relationship to chlorophyll A is encouraging though, because then you can possibly do a decent estimate of the growth efficiency and apply on other measurements, since the growth efficiency measurements themselves are pretty tedious to do. Just to mention a little bit about the key methods that we used. For the respiration measurements, we used some optoid method that we have developed in our laboratory some years ago. We tried to increase the sensitivity then by extending the incubation to 48 hours instead of 24 that we usually apply. And I think that's reasonable given the cold conditions and quite low process rates up in that area. Still, it's a chemical method, so it has some limitations in detection limit. The growth method instead was using the treated thymidine method. That's an isotope method, radioactive isotope, and really sensitive in that sense, but we still had to increase the sensitivity by doubling the sample volume, and we had 3 times as long incubation, making about 6-fold higher sensitivity on that technique, and that made the measurements to work pretty well in most cases. So quite few rejected sample in that case. So summarizing a little bit what this would mean if we now get this data published eventually. The measurements we've done of biomass growth so far, both Wheeler and us, if we use our conversion factors, would mean that much more organic carbon need to be consumed to promote that growth that we measure. But at the same time, of course, a lot of the carbon consumed is transferred to carbon dioxide rather than to biomass. Now, as I said, compared to Wheeler's estimates were about 3 times lower, but the growth efficiency is about 10 times lower. So this means in the end that combined, it may increase the use of dissolved organic carbon by prokaryotes about 3-fold. And that would mean that prokaryotes are more important than we have thought so far in terms of cycling of the carbon in the Central Arctic Ocean. Well, with that, I would need to acknowledge the master first, Mattias Pettersson, that took us through the Central Arctic Ocean and up to the North Pole again with Odin, and the expedition coordinator from the Swedish Polar Research Secretariat, Maria Samuelsson, and not least the expedition leader, Professor Pauline Snös Lejonman, that made the expedition work smoothly and in a good mood all the time. And we had a lot of support from other units too, SciLife Lab, taking care of sequencing. We had electron microscopy support. We also had funding from the Kempe Foundations and the Exchange Strategic Program, and very important technical support for Umeå Marine Science Center. So with that, I would like to thank you all for giving this opportunity and for listening.
Thank you so much, Johan. Very nice presentation, very interesting, unknown territory for my part. Do we have any questions? Well, I can start with one at least. So you mentioned that this growth efficiency is extraordinarily low.
Yes.
And I was curious about the reason, and you think because they need to for life subsistence, basically.
Yes.
They can't build much biomass. But is there anything that could change that into the future, or is it sort of species dependent, or can the individual species adapt to changing environmental conditions?
Well, a guess could be, of course, that to increase, well, either the supply of kind of good quality food from phytoplankton growth and remineralization by zooplankton, that would increase, the amount of nutrients and the quality of nutrients they need. That could improve the growth efficiency, and also a higher temperature, of course, if that would happen. And the combination of both is even better. So with climate change, if that would lead to more productivity, which you won't really know, and to a warmer surface layer, yes, that could increase the growth efficiency.
Yeah. Is there a global relationship between growth efficiency and temperature? So it's higher or is it mostly related to the quality and availability of food?
Well, I would say very bit of food, although the global compilations are not too common in that sense. Actually, it's the global relationship mostly referred to is with the bacterial growth itself. So in one sense, it shows that when the prokaryotes have better growth conditions, their growth efficiency is also higher. That's as much as we know at this point.
Okay, nice. Are there any more questions for Johan? No. Then
okay.
Thank you again, Johan. And Maite, maybe you want to inform about future webinars before we wrap up?
Yes, I can just say that we are planning for another one in about a month and a half time. We haven't set the date yet and not yet the program, so that will be announced on the mailing list and the various calendars and newsletters that are around. And then we are planning on a last webinar this year, which will be probably in the beginning of December. So 2 more webinars this fall.
Okay, so stay tuned for information. All right, and thank you to all of the speakers, Peigen, Lynn, Andreas, Rogge, and Johan Wegner for a very nice presentation. I found myself, they were quite interesting and I learned a lot. So with that, we are free to move onwards in the working day. Okay. Have a great morning, evening, day, wherever you are. Bye-bye.
Thank you.
Bye-bye.
Bye-bye.
Bye.
Thanks.